right, thank you very much. And so next, Moaz is going to MC the next session. So thank you very much, Moaz.
All right, thank you, Dr. Roth. So good morning and welcome again to the 2022 Mutational Scanning Symposium. So my name is Moaz Dawood. I'm a MD PhD student from the Baylor College of Medicine, currently interning with Dr. Leah Starita at UW. I'll be the in-person moderator. Daniel Tabbitt, grad student here at University of Toronto, the online moderator. And so my job here is to introduce the two speakers for this morning session. And so I will get to that. But before I get to that, I've been handed these three beautiful half sheets of paper, one that says 0, one that says 1, one that says 3. And I will do my best to raise it when you have 3 minutes, 1 minute, and 0 minutes remaining. All right. So to our first speaker for this session, Dr. Jacob Kitzman. So Dr. Kitzman is an assistant professor of human genetics and of computational biology and bioinformatics at the University of Michigan. Dr. Kitzman's lab develops genome-scale technologies to comprehensively identify sequence variants, resolve genomic structures, and dissect their functional impacts with respect to molecular and cellular phenotypes such as splicing, clonal hematopoiesis, and mismatch repair. His lab has extensively screened variants in the mismatch repair gene MSH2, and the title of Dr. Kitzman's talk today is Checking on the Spellcheckers, Deep Mutational Scans of Mismatch Repair Factors and Beyond. Dr. Kitzman.
All right, well, thanks so much for the kind introduction and for the opportunity to be here. Looking forward to a really great couple of days. So my disclosures. And by way of background, the genes that I'm going to talk about today underlie a condition known as Lynch syndrome. Lynch syndrome is a very prominent and, at least for me getting into this, surprisingly prevalent familial risk syndrome which predisposes to early onset colorectal and endometrial cancers. So it affects around 1 in 300 people and accounts for about 3% of colorectal cancers all told. So underlying Lynch syndrome are loss of function variants in the DNA mismatch repair pathway in one of 4 key factors that are listed here. And these follow a classical 2-hit model where most affected individuals carry an inherited loss of function variant and then have a somatic second variant later in life. And importantly, these mutations exhibit incomplete penetrance, and sort of that combined with the common nature of the sporadic cases means that many carriers for Lynch syndrome are unaware of their personal elevated genetic risk. So what do these factors do? Well, their primary responsibility is to scan the genome for mispairs which arise from replicative incorporation-based misincorporation. And they're sort of divided up into 2 teams, if you will. You've got MutS, composed of MSH2 and MSH6, and MutL, composed of MLH1 and PMS2. And these come together to scan the genome and then direct the excision and resynthesis to correct the mispairing. So a hallmark of mismatch repair deficiency is that the tumor mutational burden is greatly increased over about 2 orders of magnitude, and sort of classically and easiest to assay in its sort of classical form, this results in a form of hypermutation called microsatellite instability, or MSI. So this will be familiar to many of us, but the reason that we're here talking about this is that there's a huge VUS, or variant of uncertain significance burden. And so if you go to ClinVar, which is what I'm showing here, the great majority of missense variants for each of these 4 factors that are listed in ClinVar are VUSs or have conflicting evidence, and the converse is also true. So the great majority of the VUSs that are extant in ClinVar are missense. So there's a big problem. It's largely driven by missense variants for these genes. So we do what we do what you do, we make mutations, we try and model them in cell lines, impose some functional selection that tells us about those mutations' activity, and quantify their abundances at the beginning and to arrive at a function score. So I'll present 2 different function maps. One's published, one's not, and these are projects that were spearheaded by a former postdoc in the lab, Isaac Jia, who's now got his own group at an institute in Greater Bay in China. One sort of technical detail that was fun to figure out, due to the nature of the functional selections that I'll tell you about, we need and sort of the lengths of these genes, they're on the long side for deep mutational scanning. We need a method that is both scalable to generate the mutational libraries and has a very low off-target mutation rates. We don't want a lot of mutations outside of the region that we're trying to hit. So we developed something called cassette swap mutagenesis that entails chunking up a gene or cDNA into roughly up to 300 base pair chunks. We can then sequentially replace each of those chunks with a stuffer sequence. We can do this by hand, or these days it's nice to pay companies like Twist to do this. And importantly, we sequence verify them to ensure that our starting bits are free of off-target mutations. So we have clean backgrounds. And then we can use our favorite Gibson or Golden Gate approaches to clone in complex mutational libraries. And so when we screen a bunch of individual clones, we find that this gets rid of the roughly quarter to a third that we typically see of these types of libraries that comprise either kind of leftover wild-type clones that just get in the way and take up space, or clones with additional mutations off target that are going to screw things up downstream. And we do that for about $4 US per codon, so it's cheaper than some approaches. So we're sort of lucky within the set of target genes that I'm discussing here that we're able to kind of stand on the shoulders of some really great functional assays. So there are a variety of functional assays out there for these well-studied factors. The one that we sort of have felt is most amenable to pooled screening is drug resistance. And so the drug that we use is a purine analog 6-thioguanine, abbreviated as 6-TG. And this compound is selectively toxic to cells that are MMR proficient. So you sort of have to turn things upside down in your head. We're going to select for the loss of function when we use this. So this is an example of how it looks. We start with a knockout human cell line, and then we complement that knockout with lentivirus carrying either a wild-type MSH2 cDNA or a known, which should be functional, right, in blue, or along the bottom a known founder mutation, which is well known to be destabilizing loss of function. We grow those cells out under mock conditions. They grow at comparable rates. So MMR genes are dispensable both in vitro and in vivo for sort of cellular growth. When we treat with 6-TG along the top, that is selectively toxic to the cell where we have now complemented functional MMR, but the cells that are expressing this loss of function mutant MSH2 can keep growing. So that's what it looks like for one variant. So we did this for all the variants in MSH2. There are about 17,000 of them at the missense level, and kind of at the 10,000-foot view, we get a function map that makes sense. So we see a lot of constraint, sort of vertical ribbons of red, so red indicating deleterious variants, loss of function. Those line up fairly nicely with kind of key known structural domains, so the ATPase domain, which is essential for mismatch repair, and then a connector domain, which confers some kind of flexibility to the complex. So that was good to see. And sort of moving on from there, in more recent work, which has been led by Dr. Tony Scott, who's an MD, PhD, assistant professor in our Division of Internal Medicine, and collaboration with colleagues at Ambry Genetics, we've tried to go on to clinically validate the utility of these data. So first we did what everybody does. We compared to ClinVar. It's pretty concordant. There are a couple of outliers, who knows. But then with the power of individual tests with data that Ambry has shared, we were able to hone in on specifically 49 MSH2 missense variants. And these are selected in a particular way to exclude variants for which the standing classification relied upon functional data. Because what we don't want to do is try and validate one functional assay for the purposes of clinical application, simply using another functional assay that may be, collinear, as we've heard about, or very, very correlated. And so we can do that. There are 49 such variants. We have perfect concordance in that, relatively small set. And then kind of having done that, we can use the resulting odds path score that we sort of derived, or that we calculated for MSH2, and go on to reclassify standing VUS. And we were able to, our colleagues at Ambry have been able to reclassify 10 of them to pathogenic, likely pathogenic, and another 500 look like they could go benign. Sort of one really interesting thing, I think, that we were able to do with this for a subset of individuals who had not only germline testing, but also tumor genetic testing. We were able to look at the joint patterns of mutation in those individuals' germlines and in their tumors. So there were 25 such individuals who carry an inherited germline MSH2 missense mutation. So we go to our function map. We split those into individuals for whom the inherited MSH2 missense variant is loss of function. They're along the top. The ones that are functionally normal are on the bottom. And so kind of two striking patterns emerged that kind of diverged between the groups. So the individuals along the top who inherited a loss of function missense variant nearly universally have a somatic second hit that is pathogenic in the same gene in MSH2. So this makes sense. This is kind of the two-hit model that we expect to see. Nice to see it in practice. And then along the bottom, those individuals also have a high prevalence of mismatch repair deficient tumors. But what's different for their tumors is that they appear to be largely driven by somatic either mutations or epigenetic silencing of another MMR factor, MLH1. So sort of this type of MAVE data looks like a promising way to jointly analyze tumor and germline data. So we compared our MAVE function scores to just standing variation in the population. We find that around 1 in 57 individuals in NOMAD, if you kind of do the cocktail napkin math and just add up the allele frequencies of really rare alleles, around 1 in 57 individuals are carriers of rare missense alleles, and almost 98% of those standing missense alleles appear to be functionally normal by deep mutational scanning. And in fact, all the ones that are loss of function are exclusively doubletons or singletons within NOMAD, with the sole exception being this founder mutation that I told you about. And this is a significant depletion that we think reflects survivorship bias, and kind of similar depletion has been seen at the level of copy number variation in data sets like UK Biobank. So next we actually went and curated a slightly different kind of validation set comprised of as many reports of missense variant function as we could find out there in the literature for MSH2, so 145 variants. And we wanted to benchmark the deep mutational scanning data and kind of best-in-class prediction algorithms against that gold standard set. So, maybe not surprisingly, the deep mutational scanning does really well compared to perhaps similar lower throughput functional data from the past. And kind of promisingly, actually, 3 of the callers did quite well too, and 3 of them reached an area under the precision recall curve of greater than 0.9, which was great to see. So the deep mutational scanning data and the predictors largely agree for these previously published kind of well-known, often clinical variants. But we looked a little bit more deeply, and we were kind of surprised to see that there was quite a bit less agreement for other variants at those same residues. So that's going from now the dark blue and the dark red to the light blue and the light pink. And so in this case, I'm picking what was kind of one of the very best performing algorithms, Eve, and plotting its scores against our deep mutational scanning scores. So on the on-diagonal quadrants are variants for which the computational and the experimental methods agree. Off diagonals, they disagree. And there was kind of even less agreement across the rest of the protein. And so this is not to pick on this particular algorithm. We saw this kind of for every caller that we've looked at so far. And we think there's actually maybe something interesting here that is worth kind of digging into a little bit. So I'll focus on one example here, which is at the N-terminus of MSH2, which contains the mismatch, the so-called mismatch recognition domain, although it actually turns out that the kind of job of recognizing mismatches in eukaryotes has been farmed out largely to MSH6, so the partner within that MutS complex. But yet there's still, yet this part of the protein is well conserved, so it seems that it's doing something. It turns out that loss of the start codon, resulting in an N-terminal truncation of 40 or 50 amino acids, is not acutely pathogenic. It does not rise to the level of a Lynch syndrome mutation. And there are also relatively few non-splicing loss of function pathogenic missense variants in this domain. So it doesn't seem to be causal for Lynch syndrome to a first order of approximation, and yet it's still there. So Eve scores it very highly, suggesting that it's intolerant to mutation on the basis of conservation and coupling signals. We don't measure much intolerance there, and we're interested in sort of squaring these two, seemingly discordant observations in the hopes of gaining some insight into what the kind of function of that N-terminus of MSH2 that might be subject to kind of longer-term selection, what's actually going on there. So I guess stay tuned for that. So moving along, I'd like to kind of jump over to the other half of the team, which is in MutL, and I'll show a little bit of data on MLH1. So we've gone ahead and sort of done the same deep mutational scanning approach coupled with 6TG selection for MLH1, and this is what you get. There's kind of similarly pretty strong concordance with ClinVar And we see similarly kind of strong intolerance to missense variation at kind of known essential functional sites. So the ATPase, there's an ATPase fold in MLH1. It also does not tolerate missense variation. One thing that stood out to us is this middle region, which is kind of largely blue, and so meaning that it's kind of almost entirely devoid of missense variants that are disruptive to function under our assay, with the sole exception being a little cluster of red that you can see in the middle. Okay, and so this turns out to be a disordered linker, so it's not in any of the available crystal structures because it won't crystallize. It's well known to be disordered. It is conserved in its existence among mut-L orthologs, but at the sequence level, it's not well conserved, and its length can also vary a great deal. If you put it into AlphaFold, it suggests maybe a little bit of structure there. I don't know what that means, but I would love for somebody to tell me, perhaps in the next couple of days. And then, great work previous work from a couple of groups, including Eric Alani's group, have shown that deletion, that truncation of this disordered region of a specific subset of it is deleterious to MMR function, and that actually aligns very well with that kind of small island that we see of intolerance to missense variation. So we don't know what's going on there, but we're interested in digging into that more. Kind of one final thing that I'd like to just show the crowd here that we're also kind of scratching our heads about, but think could actually be pretty interesting, is that we go into our deep mutational scanning, we get a histogram of scores that looks like this. The synonymous are on one side. That's good to see. You can fit a mixture model or do whatever you like. But there's always this kind of shoulder in between the two peaks. Even if they're far apart and your assay is really great, we see this. And so we kind of ask ourselves, is this simply noise? If we did more replicates, would these pull farther apart and would that go away? Or might these actually reflect sort of variants that are hypomorphic, at least under the conditions of our functional assay? One thing that gives us a little bit of reassurance that they may not be just purely kind of experimental noise is that we see equivalent codons encoding for the same amino acid kind of traveling in groups. Okay, so like I've highlighted a couple of them here, but you can just visually see a lot of them on this map. Regions that so these are now individual different codon substitutions, and they're grouped by the amino acid that they encode. And so you see those groups of them that are kind of in between. They're neither red nor blue, they're white. And overall, these are about a quarter as numerous as kind of the outright loss of function mutations, fairly robustly to where you draw the line. Interestingly, and I don't have this shown here, but they don't have really nearly the same kind of pronounced enrichment for specific amino acid substitutions as we see among the outright loss of function. So it's not that they're kind of like grossly destabilizing within regions that are well-structured or something like that. They also don't appear to have a preference for any particular part of MLH1. We dug a little bit more in detail into a couple of these, and there's some anecdotal suggestion from case reports that some of these may combine in a non-additive or epistatic way. So this is one of those examples of this variant K618A, which is kind of modestly common. It's a minor low frequency of 0.3%. There are homozygotes in Nomad who do not apparently have early onset CMMRD or early onset range of pediatric cancers that would be associated with biallelic loss of function. So we can be fairly certain that these are not Lynch syndrome pathogenic variants per se, or that K618A is not. What's interesting is that has been seen, that particular variant's been seen co-occurring in Lynch syndrome families in cis with another variant, and the model has been proposed that those two together cause loss of function such that somatic mutation of the remaining wild-type copy drives tumorigenesis. So maybe pairs of these maybe the presence of K618A sensitizes other rare variants. So we're kind of starting to explore this with double mutation screens. So in this example here, we've taken K618A and R659Q. I'm showing just actually R659Q here on the plot, excuse me. So we've put that on the background, and then we've made every missense change in the region just next door to it, just as sort of an entree to look at this. And so each of the points that I'm showing here represents one of those double mutants paired with R659Q, where the loss of function measure that we get when it's paired with R659Q is shown on the y-axis. Its loss of function score by itself is on the x-axis. As a reminder, more is bad. So, you know, you get a lot of variants that don't change. So lower left-hand quadrant, variant is neutral in both contexts. Upper right-hand quadrant, it's deleterious in both contexts. And then we get a whole subset of mutants, of alleles that are loss of function only when they're paired with R659Q. So they're not loss of function by themselves, but they are when you add them into the context of that additional variant. So we think that those might be sort of individually weakly destabilizing, and sort of, you know, as we were doing this, we were inspired by some, you know, some great papers from, I think, some of the people that are, you know, in the audience and will be around here. So I'd love to kind of hear some feedback on this, but we think that these are kind of combined effects of individually weakly destabilizing variants that together kind of push them over the edge, at least within our, within the context of our assay and then possibly within the context of disease risk. What's interesting is we also see a handful, oh, and I should mention, so K618A is up in that upper right-hand quadrant. So when we do this pooled assay, we recover that as a variant that is sensitized by the presence of 659Q. What's interesting is there's a couple of variants that go in the other direction. So by themselves, they're individually deleterious. Those are on the lower right-hand quadrant. But when you add them together with R659Q, they're neutral. And interestingly, they are not simply kind of in residues that are immediately adjacent to R659Q. So, you know, this could represent an example of allostery, something that we're looking into further. So you might have noticed that that I didn't mention splicing at all. Well, I'll first just say, you know, the MMR experts tell us we need additional assays beyond just 6TG resistance. So we're thinking about ways that we can do that and have a couple of additional reporters kind of in the works. And then splicing is kind of the other thing that I haven't mentioned. So this was a cDNA-based screen. We don't capture splicing effects. But a PhD student in the lab from the bioinformatics program has been together with collaborators in Sally Camper's lab at Michigan. We've put together a massively parallel splicing assay that we think is one way to capture those types of effects. And I guess for more information, check out Kathy's poster. So just to wrap up, I've described function maps for two of the most commonly mutated genes in Lynch syndrome. We overall, between the two of those, have measurements for about 98% of the possible missense variants. And we're working towards clinically validating kind of their predictive utility. And going forward, we're really interested in surveying, kind of using Lynch syndrome genes, MMR factors, as a platform to survey intra and inter-factor epistasis between pairs of missense variants. And we're also excited to extend this type of approach to other hereditary colorectal cancer syndrome risk genes. And a postdoc from the lab, Dr. Shelby Hemker, is here and would love to have some feedback on her work looking at another gene. So with that, I'll just say thanks and looking forward to taking any questions.
Okay, so there are no online questions. Our virtual attendees are still in bed, I think. So I'm going to selfishly ask my own. So can you go into a little bit more detail on what the mechanism by which 6TG is toxic to cells that do have functional MSH2?
Yeah. Oh, no, I had a hidden slide. So it's thought that 6TG or one of its metabolites results in a lesion that is recognized by the MMR pathway. So when you kind of sprinkle that lesion all throughout the genome, it results in a futile cycle of repair that just stalls the cells or triggers apoptosis. So getting rid of MMR while, you know, subjecting the cells to kind of the mutagenic burden of that treatment, they at least don't get stuck trying to fix it. Is sort of the bottom line from what I can tell.
Thanks.
Maybe two Canadians, you know.
Sorry.
Yeah, so I live in the States, so I don't get to make Canadian jokes that often. You know, I was really fascinated by these rescue mutations in the bottom right, these suppressors. I was wondering if you looked at sort of the evolutionary dimension and whether those, you know, those changes appear in the evolutionary history of orthologs of this gene.
Yeah, that's really interesting. We haven't looked specifically for those rescue changes, but as a group, if we take the multiple sequence alignments for MSH2, and then there's actually, yeah, we haven't done this, well, no, I guess I can't quote the number for MLH1, but overall among the point variants that are, fixed in related orthologs, if they are made, if we just look them up in our data set, which is equivalent to making them singly on the human background, 98% of those are functionally neutral by our assay. So as for whether those ones are, I mean, that's an excellent suggestion. We'll go and do that right now.
So I wonder if you could speculate on, the algorithm, or the strategy for how do we choose which pairs of mutants to look for epistasis for. And, of course, you can pick the highest allele frequency functional variants, but beyond that, like, we're not going to be able to test all pairs and all proteins we care about. So how do we?
Yeah, I guess if we think that a lot of if the bulk of the mechanism is combined destabilizing effects, then maybe, predictions of stability and or, approaches like VAMP-seq or just known. I mean, for these genes, we're lucky that there are a lot of data for individual variants that are known to be destabilizing. So those would be great places to start. Or there is some interaction. There are some separation of function mutants which disrupt interactions amongst the complex members. We think those would be also perhaps interesting.
Yeah, great talk. I have a question about, you show really nice functional assays, but how about like really testing the phenotypic consequences of some of these mutations in relevant patient organoids or, PD axis, for example? Just curious about your thoughts on that.
Yeah, that would be fascinating to do. I think we don't know how to do that at high throughput, although there's a great paper from Atreya Dunham's lab that has looked at mutation rate in yeast, which is kind of like the next level, I think, of kind of intermediate readout that you'd want. And we're interested to see if we can get a readout of mutation rate in human cells, but then bringing that to organoids or other models would be great.
Hi, nice talk, Dr. Kitzman. My question is, you mentioned that you have like the neutral variants and deleterious variants, and there's hypomorphics in the middle. You are not sure if it because maybe noise by sequencing or. So my question is, when you use the 6TG to do your experiment, have you do like titration to try to figure out, and maybe the noise, maybe because of the concentration of the drug?
Yeah, we've titrated it, and we've run lower concentrations of 6TG, and as best we can tell, they are kind of equivalent, except that when you reduce the concentration, you might need a greater number of passages, but they don't seem to, using a lower concentration of drug does not bring out like a distinct subset of variants from what we can tell.
So you don't think the noise maybe because of the dosage?
We don't think the noise comes from dosage, although I think if you went to a higher dosage, some of those might disappear entirely.
Okay, thank
Yeah,
you.
Thanks, that's a great question.
Great talk. I was curious about the 6% of missense changes in MSH2 you couldn't assay. What was that a technical reason, or was there some other?
That was what did not survive our QC. So variants that are discordant among replicates, we toss them, things like that.
Okay.
All right, thanks.